Right, so chapter 1 is just going to overview various themes that we're going to be exploring this year in AP Biology. So adaptations to environment as a result of evolution is going to be a pretty critical theme that we're going to be exploring this year. Here you see a ghost plant that has been able to undergo adaptations, so it is more capable of surviving in areas that do not get significant amounts of water. Evolution is known as the process of change that has been capable of transforming life throughout Earth. Biology is the scientific study of life. Biology is not just looking at microscopic questions, it's looking at macroscopic questions, and it's also looking at how they interact with one another. So there's not a specific definition for life. What we are able to do is to recognize what living things are capable of doing. And so here we see some different aspects of living things that kind of have worked together to make those connections across different aspects of biology. We can study it again at a more molecular level. That's actually the part we're going to be focusing on in this fall semester. And then we can look at it on a larger scale. And again, that's what we're going to be looking at in the spring semester. And so depending on what we're looking at with life, we'll see different levels of organization that occur. And we will also see different properties that come out. Organelles have specific functions. Those functions within a cell may cause certain cells to be able to work more effectively in your heart as opposed to in your brain. And then even within that, we can go on a larger scale and look at it from different organisms. So large scale, we're going all the way out to the biosphere, and then small scale, we're going all the way down to atoms. Reductionism is one way we can try to manage what we're looking at studying. We can't necessarily look at something that is super complex without breaking it down, kind of like when you're working on a project. If you don't break it down into pieces, it can get a little overwhelming. Virtual learning, hybrid learning has definitely been one of those tasks. So reductionism can kind of help with this, but we also need to make sure that we understand that we aren't reducing it and having tunnel vision, that we need to look at the choices we make in the context of what else we're trying to study. Systems biology is one way we can examine a certain aspect and then be able to apply it to an entire biological system. And there's a couple of questions there that kind of show you how that could be put to use. But we're going to spend a little bit more time right now about those themes that I talked about earlier. First thing we're going to focus on is that organisms interact with other organisms. They also interact with their environment, and their interactions with the biotic factors that are present, as well as these abiotic factors, is going to have an impact on their lives. So we've seen that with global warming, and we can also see that through a tree taking in sunlight, which is able then to produce oxygen to allow other living things to be able to meet their needs. It's able to provide nutrition for animals, and then it's able to recycle nutrients that then can be used again in the future. Another thing we're going to look at is energy transfer. To be able to do various life activities such as moving around, growing, reproducing, we need energy. Energy cannot just be created out of the blue. It has to be transformed between forms. We definitely spent some time talking about that in chemistry before, and we're going to be talking about that quite a little bit, especially when we get into photosynthesis and respiration here in AP Bio. We're also going to talk about it with ecology in terms of ecosystems. Structure and function is another theme that is going to definitely predominate our study in AP biology. How things are physically structured is definitely going to influence how they are able to function. Why do we have flat leaves instead of thick leaves? Well, flat leaves are going to help to increase surface area, and that's going to help us help our chloroplasts to be able to capture light and conduct photosynthesis more effectively. Bird wings. Birds' wings are shaped in such a way and have such a composition with all the air-filled cavities to be lighter and allow the birds to be able to move more effectively. The cell. We will spend a fair amount of time about the cell in the fall. The cell is the organism's basic unit of structure and function. You can't get simpler than that and have a living thing be able to survive and reproduce. So organelles alone are not sufficient. Cells are going to have your membrane. They're going to have DNA that has all the genetic information present. We can have more complex cells, which is what we as humans have. Eukaryotic cells that have membrane-bound organelles, and they have a nucleus where the DNA resides. Or you can have very simple organisms that are consist of prokaryotic cells. They do not have membrane-bound organelles, and their DNA is not surrounded by a nucleus. It is in a generalized region within the prokaryotic cell called a nucleoid. Another thing we're going to spend time on is DNA. DNA is what all living things have in common. The chromosomes that are present in cells contain that genetic material, deoxyribonucleic acid, and the genes are what are inherited from parents, or what are inherited by offspring from their parents. DNA has to be able to duplicate itself. It has to be able to, in order for cells to reproduce. It also has to be present to be able to allow cells to continue to grow. And then sometimes there are repairs that need to take place. And so all of that in terms of cell division is essential and cannot happen without DNA. I know you spent a fair amount of time on DNA when you took your first biology course. The chromosomes, no matter how many you have, are just a long DNA molecule that have lots and lots and lots and lots and lots of genes. Your DNA is not manufactured by your parents. Your parents inherited it from their parents, and then they transmitted it to offspring. And that DNA is what's going to be able to generate the proteins that different organisms need to be able to develop and maintain their living structures, be able to do their life activities. DNA is your double helix. You've got the sugar phosphate backbone and then the nucleotide bases that serve as the rungs of your ladder. Again, the DNA is going to code for certain pieces of DNA are going to code for genes, which will be used to make proteins or will generate factors that will control the production of proteins. DNA does not leave the nucleus in a eukaryotic cell. It is transcribed into RNA, and then it moves to a ribosome that's either out in the cytoplasm free or is on the endoplasmic reticulum in the rough ER. And then that is where that piece of RNA is translated into a protein. And then in all these cases, there are various factors that influence whether that piece of DNA, that gene, undergoes transcription or translation. There's various factors that play a role. And then once those genes or the proteins have been made and modified, they get sent to where they need to go. So gene expression is just one way we can take that information from our gene and turn it into something our cells can physically use. So genomes are all of the chromosomes, all of the genetic information that's present in that organism cells. There's a lot of information present, and so there are sequencing machines that have been used to determine what are all the base pairs that are present on the chromosomes. Why would we want to do this? Remember that chromosomes are several football yards wide. Well, genomics is able to take is able to take advantage of computers being able to process things fairly quickly and look at similarities in chromosomal structure, both within species and between species. You can't do that by hand very efficiently. So that's where the computers are going to play a key role. And bioinformatics is basically the means by which those computational tools have been able to be developed to analyze that information. So since we're not just dealing with the biological side of things, we also need to have other players involved, computer scientists, engineers, mathematicians, perhaps. So it's not just a purely scientific endeavor, it really involves a multidisciplinary approach. Feedback mechanisms. Feedback mechanisms are what allow our cells to not just constantly make proteins that maybe we don't necessarily need. If you haven't had a meal in a while, chances are your body may need to get a source of glucose to be able to conduct cellular respiration. If you did just eat, then you may need to generate proteins that can help to break down that food. But if you haven't eaten, you won't necessarily need those proteins until you do. That's where negative feedback comes into play. If you have more and more of a product accumulate, that is going to shut down the series of steps, the mechanism that that product had to go through to be made, so that less product is generated until it reaches a point where none is generated. Positive feedback would be when you actually do want something to be made in greater quantities. We see this with female hormones in when an organism is getting ready to give birth to an offspring. So there's some examples of those loops. We'll spend time on those as well this fall. And I told you we'd come back to evolution, and here we are again. Evolution is what kind of connects all these themes together. We've got all of these organisms in our world that look drastically different, live in totally different environments, have different nutritional needs. What connects us all together? Organisms are modified descendants of common ancestors, and we're able to use evolution to show how there is unity among these different organisms, but also why we can be so diverse. If we have identified similar traits among organisms that may appear to be related or may not appear to be as closely related, typically we can go back and see that there was a common ancestor that most likely is where we were able to acquire that particular trait. Organism differences are going to be explained by heritable changes accumulating over time. We're going to spend more time on that in the next section. So evolution is able to look at life at all points in the history of it. So far, we've identified approximately 1.8 million. There are more and more being identified each year. It's estimated that there are anywhere from 10 to 100 million different species on our planet. Taxonomy is one way to try to organize the 1.8 million that we know about. Things that you've probably heard about in your previous biology course, domains and kingdoms are your broadest units, and then we kind of work our way down from there. Just to give you some context as to how taxonomy is changing, when I first taught biology, my very first year of teaching, domains weren't even on the agenda for us to teach to our Bio 1 kids. When I taught it maybe 5 years later, we went from 5 kingdoms to 6. Or maybe back then it was, I think it really was 5 kingdoms at that point. We went to 6 kingdoms, and then we added on the domains. So this is still in flux as we acquire new information. There is still plenty of debate as to how this organization should go. Right now, with the 3 domains, the bacteria would be your kingdom bacteria. Your domain archaea would have been your archaea bacteria, and those are your prokaryotes, your single-celled microscopic organisms. The eukarya domain is going to have your plant kingdom, your fungi kingdom, and your animal kingdom. When the last time I taught Gen Bio, we had a protist kingdom. The protist kingdom actually is no longer thought to exist. There are too many differences within the protist kingdom. They are a part of the eukarya domain, but they no longer feel that that kingdom should exist on its own. And they being the scientists that put together all those taxonomic categories. So how we're able to see that all of the living things that exist and how diverse they are, what they have in common, is going to be through the DNA. No matter whether you're a flower or whether you're a human or whether you are a prokaryotic single-celled organism, we all have DNA. It is universal throughout with the same A, G, C, T bases. We can also see, even though we may have very disparate physical structures, that there are similarities, maybe on a much smaller scale. Here we see we've got cilia in Paramecium, a protist, and then we've got cilia in windpipe cells. And although those are vastly different organisms that those cilia are coming from, when we take a cross section of them, we see that they pretty much look exactly the same in terms of their structure. So the idea of evolution first came about from Darwin. Fossils and other evidence has been used to document life's evolution over billions and billions of years. Darwin did his travels. He went to the Galapagos Island. He made his observations. He eventually published on the origin of species by means of natural selection. And there were two big points that he made: that species show evidence of descent with modification, that their genetic material is being transmitted from parents to offspring, and that natural selection is going to influence, is the mechanism behind that transmittance. His theory was able to connect together both the unity component as well as the diversity component. So some observations that Darwin was able to make: that within a population, we aren't all identical, no matter what population of organisms you're talking about. The traits that we vary in, there are many that are heritable, that are transmitted from parents to offspring. Survival of the fittest, unfortunately, is at work. There are more offspring produced than often survive. So competition is going to play a role for resources to be able to be successful and to survive and reproduce. The species that are present are typically going to be ones that suit the environment in which they have been placed. From those observations, he felt that the species that were present in their environment, that were best suited for that environment, were more capable or more likely to be able to survive and to reproduce. And the ones that weren't as likely to survive and reproduce were not as well suited. They were at greater risk for being crowded out for resources. So that over time, although the genetic material had not changed, the ratio of alleles present in that genetic material might have increased in terms of amounts, that you would find greater amounts of the alleles that would allow that particular organism to survive in its environment. So if there's an increased probability of having those favorable alleles, you're going to have more individuals end up with them. Because of this competition for resources, because of the unequal reproductive success of individuals, populations' alleles are going to change in frequency. The environment is going to play a role where the resources are that allow those individuals to be able to survive. So Darwin felt that when the environment was selecting for the propagation of traits that would allow those individuals to be able to survive, that this was called natural selection. So that organisms will adapt to their environment to be more successful in it. Here we've got an example with different colored beetles. The colors again are from inherited traits. We see again how you have something in your environment reducing individuals that have a certain trait, one that is easier for them to see. So the more favorable trait, those beetles are more capable of surviving and reproducing. That's going to change the frequency of the alleles that are present in those varied inherited traits. And so over time, we see fewer and fewer of the brightly colored beetles and more of the darker color, the ones that are able to camouflage in that environment. So talked about fossils a little bit earlier. We can also see how we can be, although there can be a great deal of diversity within living things, we can see how there are similarities in traits that have been passed down over time. Bat, human, horse forelimbs, whale flipper have a very similar skeletal architecture that could not be determined without looking at their fossils and looking at the similarities between them. So this was another piece of evidence that kind of backed up where Darwin was coming from. He proposed that because of natural selection, you could have a common ancestor, an ancestral species, that was capable of giving rise to two or more descendant species. And that's where the Galapagos finches are going to come into play. And you can show relationships with tree-like structures, tree-like diagrams, which I'll show you on the next slide. So there you've got your common ancestor, and you can see how the common ancestor has a connection with all of these finches over here on the far right. The finches that are more closely related are going to be the ones that are on branches of the same size that are closer together. You can set up these with fossils. You can set these up with DNA. You can set these up with observations. That's why there still are lots of debate about these evolutionary trees, depending on what people are using as their information source to kind of show these relationships. But regardless of what you're using to show these evolutionary trees, they do help us to see what species are more closely related and what species kind of were evolved over time from their common ancestor, either more frequently or more recently or much further away. So this next section is just a review of experimental design. Science means to know. Inquiry is looking for information. Scientific process is going to be involving observations, hypothesis, and then actual experiments. So biologists will describe both natural structures and processes. Y'all are going to be doing this a lot with your field experience. We collect data. Data can be collected either in a qualitative manner or quantitative manner. Qualitative would be more descriptions, observations. Quantitative would be measurements. Inductive reasoning is taking those observations and using them to kind of work backwards to form conclusions. Okay, so taking observations and trying to put those together to come up with a larger idea. Hypotheses can come about from those. So hypotheses can lead to additional observation or they can lead to actual experimentation. And there's an example with your flashlight. Okay, your flashlight doesn't work. Why doesn't it work? Here are a couple hypotheses, and then we can test those hypotheses out. When we're testing hypotheses, we're using deductive reasoning. So we're putting a question, we've decided on a question, and we've put together a plan to test what we feel is going on with that based on our hypothesis. Just because a hypothesis is supported does not mean it is proven. You can support a hypothesis or you can disprove a hypothesis, but that doesn't necessarily mean that by running your experiment and getting data that supports it that you have absolutely proven it. Hypotheses have to be both testable and they have to be falsifiable. They have to be able they have to be able to be disproved. If we are dealing with supernatural explanations or religious explanations, we aren't dealing with science. We aren't dealing with something that is testable and falsifiable. So explanations outside of that are not going to be examined in this class. Scientific method does not, there are definitely key aspects to it, but there is not a set pattern that you have to follow. It's not a cookbook. You are going to use what you have to decide which path you want to take and how you want to go along that path. An example we're going to look at is snake populations mimicry. Many poisonous species tend to be pretty brightly colored. That kind of warns off potential predators so that they aren't harmed. Mimics are going to look a lot like those harmful species, but they are harmless. So Henry Bates hypothesized that mimicry evolved in harmless species as an evolutionary adaptation to increase their chances of survival, to prevent them from being eaten. So his hypothesis was tested with the venomous eastern coral snake and its mimic, the non-venomous scarlet king snake. Both of these you're going to see on a map shortly live in the Carolinas, but there are regions where we find the king snakes without the venomous coral snake. So if predators, in theory, inherit an avoidance of that coral snake's coloration, the colorful king snake, the one that is not venomous, is going to be attacked less often where we see the coral snakes that are venomous. So there's your map. You can see the overlap. You can see the area where you only see the scarlet king snake as well. So to test this, researchers made lots and lots of artificial snakes, ones that looked like the king snakes and ones that just were plain and brown. Equal numbers, again, we want to try, when we're performing experiments, we want to try to minimize variables. Equal numbers were placed at different field sites, including areas that did not have these poisonous coral snakes. After a period of time, scientists retrieved the snakes, counted the bites or claw marks, and the data that they gathered did fit the predictions of the mimicry hypothesis. The ring snakes were attacked much less frequently in the areas where the coral snakes were. So the coral snakes' coloration was kind of assigned to the predators to stay away. There's your artificial snakes, and you can see the ones that were attacked, the brown one, and the ones that weren't. Okay. Now, when the coral snakes weren't present, the bright color did cause the artificial king snakes to be attacked. So the brown snakes did have a little bit of protection when they were in an area where the coloration did not indicate there was going to be a concern for potential predators. But when the coral snakes were present, the coloration of them kind of warned off potential predators and they went after the brown snakes instead. So when we're performing experiments, whether we're dealing with it on a microscopic level or a macroscopic level, we want to be able to compare experimental groups to control groups. We want to minimize variables. We want to, if we're going to have other variables, we want to try to keep them as constant as possible. And we need to be able to reproduce our results, to be able to verify the observations that we've obtained. Theories are definitely broader than hypotheses. Theories are based on multiple tested hypotheses. There's lots of evidence to support theories, but just like with hypotheses, if we gather new information, new evidence, theories may need to be modified. So science in general, not just in biology, but science in general, benefits from working together cooperatively and from having different perspectives. Working in teams, getting different viewpoints on how an experiment is going, or having people look at a question from different ways can sometimes lead to a path that we weren't expecting. Being able to share that information, being able to communicate it is also going to play a role. Checks and balances, having others check on one another. We're seeing a lot of this right now with all the COVID research that's going on. There's been a lot of red tape cut so that scientists all across the world can share their information with one another to try to help us come up with good approaches to attack COVID-19 to try to address this pandemic. Scientists will share their data with each other. They'll check each other's data. They'll also share it regarding model organisms that they might be using with one another. Science's goal is to look at how natural phenomena work. What do they do? How does it make sense? Technology is taking that knowledge and being able to apply it for a specific reason. So they are connected with one another, but they are distinct from one another. One example of how science and technology have worked together, Watson and Crick figured out about DNA, the structure of DNA, and then technology has been able to be developed so that we can analyze DNA on very small scales so that we can look at an individual's DNA to try to identify if there are genetic diseases that are being passed through families. We also have a lot of ethical issues we have to watch out for when it comes to technology. There may be hereditary diseases that can be identified that might cause you to decide to not have additional children. But those are choices that you need to be making, that individuals need to make. The information is there for them, but they also shouldn't have that information held against them. Having different cultures play a role, having different ideas be a part of this is also going to be really critical. Just having different perspectives in general is going to be beneficial in pretty much any situation, but especially in science. You're going to get different perspectives, and that can help you maybe look at a question in a way that you might have not have seen it otherwise.